Hey, everyone, welcome to the CCMB seminar series. It's my pleasure to welcome Sriram Chandrasekaran. Sriram is joining us as an assistant professor in biomedical engineering. He's also has a new appointment in CCMB. He just started in January. He received his PhD from UIUC in biophysics and computational biology. And then he was a junior fellow in the Society of Fellows at Harvard, where he did research on metabolism and regulation. In his lab here, he still maintains a focus on systems biology and drug discovery, which we'll be hearing a little bit about today in his talk, Metabolic Networks in Space and Time. Thanks for joining us.
Thanks, Alan, for the invitation, and it's great to be here in CCMB. So today I'm going to talk about some of the research my lab is doing on building computer models of biological networks. And specifically, I'm going to talk about new algorithms that we have developed for understanding cellular metabolism, starting with bacteria all the way to stem cells. So the modeling work we do is what's called genome-scale models. We start at the most fundamental level in the cell at the level of individual molecules like RNA, metabolites, proteins, and we try to understand how these fundamental molecules interact with each other to form protein complexes or metabolic pathways. And then we try to build networks that try to understand how these pathways interact with each other. So we have like metabolic network that characterize all the metabolic changes that happen in the system. We have regulatory network that tries to understand how different molecules regulate cellular processes. And something unique about our lab is that we also build what's called multi-network models that try to understand at the network level how do different processes and different networks interact with each other so that we can start with molecular level changes and try to predict system level changes happening at the cellular level or even at the organism level. So I'm going to give examples throughout the talk where we start at the molecular level, figure out what happens when there's a genetic mutation or change at the metabolic level, and how does that impact the entire cell or system. And we can then use these models for either drug discovery or for engineering a cell or even for engineering stem cells for regenerative medicine. So how do you build these models? So we make use of diverse kind of data. We do a lot of text mining to mine literature to figure out what's known about a system biochemically. We also use genomic information to figure out what genes are present in an organism, and then we use transcriptomics data and proteomics data to figure out what are the levels in which these genes are active in a system. And then we then develop algorithms that can, in an automated fashion, take these high throughput data and then combine them to build these genome-scale network models. So ultimately, all these models in the computer becomes a matrix, and this matrix we call as a stoichiometric matrix. So almost every cellular process can be represented as a biochemical reaction. So it could be canonical metabolic pathways, or it could be drug transport, or it could be even DNA replication. Almost everything involves interconversion of metabolites and involves consumption or release of energy. And so we build what's called a stoichiometric matrix that represents, for example, what metabolites are being consumed in a reaction, what are being produced, and what is the stoichiometry. So almost every process we integrate becomes this big giant matrix. And once we build this giant matrix, we then make sure that it follows fundamental physical and chemical laws. For example, we make sure that there's conservation of mass. That means when a chemical transformation is modeled, we make sure that there's no net carbon balance or nitrogen balance. So there's no fundamental violation of thermodynamic laws. And we also apply biophysical constraints so that make sure that we sort of get like a feasible solution space for the network that is possible within the realms of thermodynamics and biochemistry. So these are some of the models that we build look like. So here's an example from human metabolic reconstruction. So this has more than 7,000 reactions. So this could be canonical metabolic pathways. It could be even DNA replication or transcription. And we also have more than 2,000 metabolites, more than 1,700 metabolic genes. And these genes encode more than 2,600 metabolic enzymes. We also have various compartments in the model. So for example, glucose in the cytoplasm is very different from glucose in the nucleus, and it's different from glucose in the mitochondria. So we have compartments within our model. And for example, this model captures the interconversion of metabolites and what enzymes convert these metabolites, and then what genes encode proteins that form these enzyme complexes, and how these enzymes are then organized into pathways, and how these pathways fit into the entire cellular network. So we can model all these changes using this model. So these models sort of represent like a biochemical map of all the metabolic transformations that happen in a system. And the way we use these metabolic models to simulate them is very analogous to how we use Google Maps. So when you as a user use Google Maps, you provide two inputs to Google. One is your objective, that is your source and destination. So you want to say that I want to go from point A to point B, but then that alone is not enough for Google to figure out the most optimal route. Then you provide constraints. So your constraint could be I want to minimize time, or you want to minimize cost, or it could be minimize distance. So depending on the constraints and the objective, Google provides you a unique solution among millions of possible answers. Similarly, in a metabolic map, we can also use similar ideas to identify optimal metabolic states of a cell. So, for example, just like in Google Map, you have an objective. For a cell, you can imagine the objective of a cell would be to produce more of itself. So you can write this objective quantitatively. So, for example, a typical bacteria needs to produce various amino acids, nucleotides, lipids, and you also need to produce it in a proper proportion to produce more of itself. So we call it the cellular objective, and it needs to produce these components from nutrients that are present in the media. So it could be glucose and urea, or glucose and amino acids that's present in the media. The cell then consumes these and produces all these components, which is its objective. And then we have constraints. Just like in Google Maps, you have time and distance constraints. Similarly, in metabolic map, you can use constraints like figure out the most optimal route that is most thermodynamically efficient. Or you can ask, say, another constraint, which is let's achieve this objective with the least number of enzymatic steps possible. So depending on these constraints, the algorithm can figure out the most optimal route from input nutrients to the output and figure out what reactions are most likely to be active in a system. So using this now, you can ask complex questions like what happens when I knock off a gene, or downregulate an enzyme, or inhibit a protein. So or even combination of proteins. So for example, here, let's say I knock off a gene in this model. And so let's say this gene encodes for a single protein, which then catalyzes a single reaction. So then what you would say is that we'll knock off this reaction in the matrix, and then the model will predict what happens to the cellular objective when this gene is knocked off. So you can imagine three different possibilities. So if this gene is very important and it produces an important amino acid required for achieving the cellular objective, then the model will output saying that this gene knockout is lethal to the organism. But what's normally the case is that the algorithm might find an alternate route through the metabolic network, and in that case, the algorithm would say this gene knockout does not have any effect. Or there's a third possibility, which is that when you knock off this gene, it uses a different reaction, but that reaction consumes more energy, and so it's a less efficient route for the model. So the algorithm might say the cell is viable but grows at a more suboptimal route. And it can also output saying, for example, this gene knockout will result in consumption of a different metabolite and secretion of a new metabolite, which you can experimentally verify. So these models are like really powerful way to either engineer a cell, or you can find metabolic vulnerabilities of pathogens. But one of the main challenges right now in this field of modeling is that these models sort of represent like a huge data bank of all known biochemical knowledge of a system. So we have a model for humans, we have a model for mouse, but the main challenge right now is we don't know what sort of reactions are active in a cell or a system at any given point. So for example, if you think of human cell or muscle cell, even though the model has 7,000 reactions, only a few hundred are going to be active in a muscle cell, maybe a few thousand are active in a liver cell, but we don't know that just by looking at these models. So the main challenge right now is building what's called context-specific models. Context-specific means what's the network activity of a given cell at a given point of time. And so most of my work has been trying to develop algorithms or methods for creating these context-specific models. So we take these big giant networks and figure out which some of them among some of the reactions in this giant network are active and what is their activity at any given time point. So for the rest of the talk, I'm going to give examples of how we've used metabolomics data, proteomics data, and transcriptomics data, and we have used these different data sets to create context-specific models in different examples. I'm going to first talk about our application in stem cells. We also talk about work in pathogens and also on immunology. So let's talk about integrating metabolomics data with these models because it's the most intuitive. So we usually developed a new method for combining metabolomics data with these models. So traditionally what people do is that when they measure metabolomics of a system, they would use the presence of a metabolite as and additional evidence supporting the presence of a reaction or an enzyme. So what we have developed is a new method that can extract more information from metabolomics data. So we can not only say whether a reaction is present or not, but we can also say how active reaction is compared to another system. So anyone who's worked on metabolomics data knows that it's interpreting this data is extremely challenging. You normally, this is like a typical example of metabolomics data set where you're comparing two different conditions or two different cell types. You can see there's a lot of metabolites that are changing. Some metabolites in the T cycle are going up, some metabolites are going down, some nucleotides are increasing, some nucleotides are decreasing. Just by looking at this data, it is very hard to say what pathways are more active or what is the causal change that's driving these metabolic changes. And so people have tried many statistical approaches. So one common way to interpret metabolomics data is by doing what's called pathway enrichment analysis, where you would overlay this data onto predefined pathways. But there are two main challenges in doing this for metabolism. First is that let's say miraculously all metabolites in T cycle are increasing in your data set. Even if that's the case, we do not know if that change that you observe is either due to increased activity of the TCA cycle, or it's maybe something due to the fact that pathway downstream of TCA cycle is less active, or it could be that pathway upstream of TCA cycle is more active. So pathway enrichment analysis cannot tell you or resolve all these differences. So to address that, we developed a new method that makes use of these powerful networks, and we overlay these metabolomics data onto these to identify metabolic features that are most likely to drive the changes that you observe. So I start with a simple example. Let's say you just have two measurements. Let's say in your data you observe increased production of lactate and increased consumption of glucose. So and then you overlay that onto the model. The algorithm would output saying that increased glycolysis is probably the most likely explanation for the observed metabolic data. So if you have 100 such data points, the algorithm will find the most optimal metabolic state that is consistent with all the entire set of metabolic observations that you made. So then if you have two different conditions, then you can overlay metabolomics data from condition 1 and 2, and you can create two different metabolic network models, and then you can compare those and figure out what genes or metabolic enzymes are most likely to explain the differences that you observe between these two data sets. So mathematically, this is how we do it. So we make use of time course metabolomics data. So we don't use steady state snapshots, but we need time course changes. So we assume that the changes that you observe using time course data is the net result of the fluxes that are either producing or consuming a metabolite. So let's say we have observed lactate is being secreted more. We assume that it is the net result of reactions that produce lactate and reactions that consume lactate. So in the model, there may be 100 such reactions that do so, but then we know what the net result of the fluxes of these reactions are. We know from the data that lactate should, in the end, be increasing over time. So then if you have 100 such measurements, the algorithm will then figure out what is the most optimal fluxes that best fit the observed data that you see. So another thing is that given that there's a lot of noise in these measurements, we try to qualitatively fit the experimental observations. So for example, let's say you observe glucose is increasing at 10 units per hour. The algorithm will try to fit the trend that glucose is increasing. It won't actually fit the exact value. And another main reason is that when you do metabolomic measurements, you do it from bulk cells. Whereas in the model, you have 8 different compartments. And so we don't know from which compartment is dominating the signal. For all those reasons, we just try to match the trends that you observe from external data. We don't directly fit it. So to test this approach, we apply this to a panel of cancer cell lines. Basically, we use the NCI 60 panel, which has 60 different cancer cell lines from breast tissue, lung tissue, and CNS nerve system. And so we used data from Jain et al., where they measured metabolomics at 2 different time points from these cell lines. So this is an extracellular metabolomics data, but the algorithm can use both extracellular and intracellular. So using this data now, we can create 60 different models for each of the cell lines. So now we can ask, what are the metabolic differences between these cell lines? And can the approach accurately recapitulate what's known between these different cancer cell lines? So what we did was we knocked off every single gene that is present in these cell lines. So I mentioned before that we can do gene knockouts and then figure out how the gene knockout impacts the cellular objective, that is the cell's ability to produce nucleotides, amino acids, and all the biomass precursors. So then we can figure out what genes are most important for a cell type and what are not. So by doing this, we found genes that are lethal. That means they prevent the cell from producing biomass across all cell lines. But we also found gene knockouts that were selectively lethal. That means they were sensitive to one cell line versus others. That means we can identify really specific metabolic vulnerabilities in one cell state versus the other. So this is like a snapshot of the external data, data that we generated. For example, most of the gene knockouts do not impact cell growth. So you have a growth rate of 1, and some impact growth of all cell lines, and some show an intermediate trend like I mentioned. So how accurate are these predictions? So to test this, we compared the model predictions with data from CRISPR-Cas9 and siRNA screens. And what we found was that metabolic genes that were predicted to be selectively essential by our algorithm were also more likely to be selectively essential in the CRISPR-Cas9 screens and also the siRNA screens. So this suggests that just by using metabolomics data, we can say something about what are the most important metabolic genes for a specific cell line or tissue. So it's not a perfect overlap. This is because both the data sets that we used are highly noisy, and also the metabolic conditions are slightly different. So the fact that we are still seeing a very good overlap is still heartening. So then we next applied this to understand stem cell metabolism. So just a brief background, what's so interesting about stem cells is that there's been an increasing interest in understanding stem cell metabolism, mainly because it's been found to not only impact the growth of stem cells, but also their ability to differentiate, and also their pluripotency. By pluripotency, what I mean is that stem cells have a unique property, they're able to give rise to various cell types, and metabolism seems to play an important role in maintaining the pluripotency of different stem cells. Another interesting observation regarding stem cell metabolism is that distinct phase of development, starting from your single cell embryo all the way to the organism, has distinct metabolic requirements. So for example, here you can see that during the one cell stage, the embryo seems to consume more pyruvate, and as they differentiate more, they seem to consume more glucose, and then there is also a differences in their respiration rate depending on the development state. So there's a lot of interesting metabolic differences has been known during development, but most of the studies so far have only focused on glycolysis or TCA cycle, which are just 2 or 3 among the hundreds of pathways in a cell. So we want to take a much more systems level perspective and try to understand what are the entire set of metabolic changes that are happening during stem cell differentiation. So as a model system, we looked at 2 distinct stages of development. It's called the naive and primed stem cells. So to orient you, we have the one cell stage, and then the embryo divides and you get like a bag of cells, and we call the blastocyst. And there's the inner cell mass of the blastocyst. That's where you derive your embryonic stem cells from. So the naive state represents these embryonic stem cells, which can give rise to all cell types of the body. So the prime cells are sort of slightly more advanced than naive cells in embryonic development. So for example, for mouse embryonic development, the naive cells represent around day 4, and the prime cells represent around day 6 of embryonic development. And the prime cells are considered to be the cells where they're getting ready to differentiate and give rise to other cell types. And so understanding the metabolic differences between naive cells, which are highly pluripotent, and prime cells, which are getting ready to differentiate, would tell us what are the metabolic differences underlying stem cell pluripotency and what makes a cell a naive cell versus a prime cell. So then we can use this information to even engineer cells towards a naive state. So we did metabolomics on these different cell types, and this is the data that I mentioned to you earlier, where we saw a lot of metabolic differences, but it's very hard to tease out what are the pathways that are different between naive and prime cells just by looking at this data. Also want to mention that this was done in collaboration with the George Daly lab in Harvard Medical School. So we did metabolomics on 3 different time points so that we can use that with our approach. And we measured around 200 different metabolites. So we went ahead and integrated that into our model, and then we created a naive cell metabolic state and a prime cell metabolic state. And just like the cancer example, we then knocked off all genes and reactions in the naive metabolic state and the prime metabolic state, and we tried to figure out what are the differences between them. So majority of reactions, they're similar between them. So this histogram shows the distribution of metabolic differences sort of, and you can see that majority of reactions when they're knocked off do not are sensitive in both naive and prime cells. But we found a handful of reactions that are predicted to be more sensitive in prime cells. That means knocking off that gene is important for the prime cell's metabolic functions. Whereas we found another set of genes involved in oxidative phosphorylation and nucleotide synthesis, pyrimidine synthesis, to be more sensitive in naive state. To orient you on what these pathways are, so in the prime cells, we saw an increased activity in what's called 1-carbon metabolism. So 1-carbon metabolic pathway branches off from glycolysis, and it's involved in serine synthesis, glycine synthesis. It's found in production of folate, which is needed for purine synthesis. And it's also required for producing methionine, cysteine, and it's also needed for producing acetylinosyl methionine. And this is needed for DNA methylation, histone methylation. So it's a very important pathway for the cell. Whereas in naive state, we found increased activity of glycolysis, oxidative phosphorylation, and the pentose phosphate pathway. So to test these predictions, these sound interesting, but to validate these, we used metabolic inhibitors, and then we treated both naive and prime cells to these metabolic inhibitors. So if the model predictions were correct, then we should see that in the prime cells should be more sensitive to inhibition to the folate pathway. And so we used an inhibitor called methotrexate, which inhibits the dihydrofolate reductase enzyme. And what this graph shows is an increasing dose of this drug, methotrexate, and we measured cell viability. And you can see that the prime cells were more sensitive than the naive cells at different doses of this drug. So this means that the prime cells are more sensitive to inhibition to folate inhibition, which validates the model predictions. Another really interesting prediction the model made was that even though the folate pathway was predicted to be higher in the prime cells, there's a small set of reactions involved in mitochondrial folate metabolism that are predicted to be more active in naive cells. So this is very interesting because when you do metabolomics, you do it from bulk cells. There is no information on mitochondrial metabolism or cytosolic metabolism. But the algorithm was able to tease that out from the topology of the metabolic network. So we want to test this prediction. And so we used a drug that is highly specific to mitochondria. So we used a mitochondrial folate inhibitor called MTHFR2I from Race Therapeutics. And what we found in this case was that the naive cells were more sensitive now to increasing dose of this drug than the prime cells. So which once again was validates our model prediction. So just to further drive home the point that these predictions were due to the difference in metabolism, not due to some side effect of these drugs, we did what's called 13C flux tracing. So in this case, what you do is you take radio-labeled glucose and then feed that to cells. So and then you see where the radio-labeled carbon is getting accumulated in different cellular metabolites. So the model predictions were correct. If you feed in radio-labeled glucose, you should see the 13C carbon going to serine, glycine, and other 1-carbon pathways in the prime state. Whereas you should see the glucose going into PCA cycle and oxidative phosphorylation in the naive state. This is exactly what we see in this heat map here. Red is high, and prime cells are shown here, and naive cell data is shown here. You can see in the prime cells, radio-labeled glucose goes into serine, methionine, and phosphoserine, like the 1-carbon pathway. And whereas in the naive state, we see glucose accumulating into citrate, malate, and ribose phosphate. So in the TCA cycle and pentose phosphate pathway. So which is consistent with what the model predicted. So another interesting question is, why is this happening? Like, why is the metabolic rewiring happening in the prime cells? So one idea is that naive cells are considered to be epigenetically flexible. So these are embryonic stem cells. They can give rise to all cell types in the body, and they have very flexible epigenome. Whereas the prime cells, during that stage, there is pervasive histone methylation that happens. And so the substrate for histone methylation is acetylinosyl methionine. So we thought maybe this metabolic rewiring is happening to produce this important molecule acetylinosyl methionine, which is needed for histone methylation. So to test this, we then went back into the model and then identified metabolic reactions that may impact the production of acetylinosyl methionine. And so what we found was that almost every single reaction that can impact acetylinosyl methionine production was more sensitive in the prime cells. So you can see here that every single reaction that we knocked off was more sensitive in prime, but none of them were sensitive in naive state. So what this shows is that in the prime metabolic state, is rewired to produce more S-adenosylmethionine, which then impacts histone methylation in the prime cell. So if that was the case, if you perturb this pathway, then that should affect the prime cells more than the naive cells. So we used another metabolic inhibitor called DZA. So the DZA inhibits the production of S-adenosylmethionine. And what we observed was that when you looked at three major histone methylation marks, H3K9, H3K27, H3K4, we saw that methylation in prime cells were decreasing considerably compared to naive cells. Another interesting observation that we made was that the methylation in naive cells actually increased. So what the model predicted was that prime cells should go down. This was not predicted by the model, the increase in naive state. We don't know why that's happening. It could be due to some kinetic effects of drug inhibition. We are not sure, we are not able to explain this observation. So another application of this work is that since we're able to accurately characterize the metabolism of different cell states, we can ask the question, can we use this for engineering cells or reprogramming cells? So for example, normally when you want to reprogram stem cells, what you would do is, let's say you want to convert cell type A to cell type B, you would then look at some specific markers of cell type B, and then you would say, okay, I see these 5 markers, I think my programming engineering effort has worked because I see these 5 markers. But then using our approach, you can now look at 7,000 reactions and then ask the question, is the cell that is very similar or different to what I was expecting? So we can use this to assess cellular state using this algorithm. So you want to test this idea by using what's called Lin28. Lin28 is a important pluripotency marker or factor that is required for naive cells to become prime cells. And if you take induced pluripotent stem cells and knock them off, knock this gene Lin28, then they're supposed to be frozen in a more naive-like state. So it's sort of like engineering the cells to be more naive-like. So we want to test this where we chose 2 different conditions where we compared cells with intact Lin28, which can progress to prime state, and those in which Lin28 was knocked off. So they're more in a naive-like state. And then we did the metabolomics again, and then we tried to see what are the metabolic differences between Lin28 knocked off cells and wild type Lin28 cells. So just like before, we found very similar differences between the prime cells with intact Lin28 and naive cells with Lin28 knocked off. Once again, you can see that there's difference in oxidative phosphorylation, pyrimidine synthesis. In the prime state, we saw differences in folate pathway and furin synthesis, just like earlier. But you also discovered some nuanced differences. If you are paying attention, one thing I mentioned earlier was that the naive cells, the pentose phosphate pathway was higher, like in this figure. Whereas here in this case, we saw prime cells had higher pentose phosphate activity. So what this suggests is that knocking off Lin28 can freeze cells in naive-like state, but these cells are not exactly identical to the canonical embryonic stem cells that we saw earlier. So we can also look at cellular states from a more 10,000 foot perspective. We use what's called multi-dimensional scaling that summarizes the entire set of differences that we saw across 7,000 reactions. So in this plot, distances represent how similar or different cell states are. So for example, you have the naive state here, you have the prime state, and then you have the wild type cells with intact Lin28. And knocking off Lin28 is supposed to make cells go towards a naive-like state. This is exactly what we see. Cells knocked off of Lin28 move closer towards naive state. But then these 2 dots are not on top of each other because we saw some nuanced differences in pentose phosphate path. So we can use this to characterize different cell states, figure out how similar or different they are from a metabolic perspective. To summarize what I've said so far, we have talked about a new method where we can overlay time course metabolomics data with these genome-scale network models, and then can identify nuanced differences between different cell states. And I talked about how we found differences in the 1-carbon pathway and how that ties to histone methylation and what makes a prime stem cell different from a naive stem cell. So going back, so I first talked about how we can overlay metabolomics data with these models. So I went into a lot of detail on the modeling for this example, but for the next 2, I'm just going to talk about the main take home that we found by doing this modeling analysis. So the example of the proteomics that I'm going to talk about a recent work that I did in collaboration with Judy Lieberman's immunology lab at Harvard Medical School. So we were interested in studying a new mechanism by which immune cells were predicted to clear pathogens. So killer T cells play an important role in your body. They play an important role in clearing cells infected with bacteria, viruses, and they also play an important role in clearing cancer cells. And so we were interested in a new mechanism by which these killer T cells are clearing these bacterium infected cells. So normally this is how they do it. So killer T cells secrete what's called perforins and granzymes. So perforins sort of form a hole in the target cell, and they deliver these uh, enzymes, granzymes, which then are supposed to cleave proteins in the target cell to cause cell death. But what we do not know is how the cell death actually happens. Does this enzyme cleave randomly across all proteins, or is it more like an antibiotic where it goes directly towards cell wall or ribosome and then degrades the ribosome? So we don't know how the cell death mechanism actually happens. So we want to take a proteomics approach and use our modeling to figure out, understand the mechanism behind how this immune clearance works. So understanding this can help us design new antibiotics. So let's say we discover that a new protein is being targeted, then we can design new drugs to target that protein. That could become a new drug for treating drug-resistant infections. So to understand the mechanism behind how this works, we did proteomics on 3 different bacteria, E. coli, tuberculosis, and listeria, and we exposed this to these granzyme proteins. And then we did 2D gel electrophoresis, where you expose it to these granzymes, and then you have a control lysate. And then by using electrophoresis, you can separate out proteins that are being cleaved by these granzymes. We then did mass spec on these spots to figure out what these proteins are. So we then overlaid that onto a model, and what we found was that there are a lot of different kinds of proteins that are being targeted by these enzymes. So we saw proteins all over the cellular map. I want to focus on the ones in red. These are targeted in more than one pathogen, and these were like canonical pathways like TCA cycle, glycolysis, oxidative phosphorylation, fatty acid synthesis, and degradation. So a lot of pathways were degraded. So we want to know, the first question was, is this enzyme randomly degrading proteins, or is it going after targeted enzymes? So we then simulated the impact of a random protease, and we compared that with the actual targets of enzymes. And what we found was that the actual targets were very different from an activity of a random protease that would degrade either based on the abundance of proteins or just highly conserved proteins. So what this suggests is that granzymes are going after some important key nodes in the network. So we want to ask the next question: what happens when you downregulate or inhibit these enzymes, and how does that impact the bacteria? So one cool thing what we found was that when we did the modeling where we downregulated these enzymes, we found that the bacteria was unable to grow in over 125 different conditions. So this is interesting because most of the pathogens in the body have a very flexible metabolic network. So when you normally grow bacteria in culture, you usually give glucose or amino acids to grow. Whereas inside the body, the bacteria is able to grow in using a variety of nutrients. For example, MTB, the bacteria that causes tuberculosis, can consume fatty acids, cholesterol, carbohydrates. So it has a very flexible metabolic network. So if the enzyme just went after one metabolic pathway, like glycolysis, then the pathogen would just use a different pathway to produce energy. And so by targeting hundreds of different pathways, it's preventing the bacterium from surviving inside the cell. So another interesting thing is that if we now look at how conserved these targets are, so we went back and looked at more than 300 different microbial genomes, and what we found was that the enzymes that are being targeted, these bacteria were also highly conserved across the evolutionary tree. And it suggested that the enzymes that are being targeted by these granzymes were also predicted to be present in the last universal common ancestor. So that means the granzymes are trying to go after highly conserved but also highly essential metabolic enzymes that prevent the bacteria from surviving in a diverse set of metabolic conditions. And so it's not targeting proteins specific to tuberculosis or E. coli, but it's going after highly conserved metabolic enzymes. And many of these are not being targeted by existing antibiotics. And so our next step in our lab is to figure out which of these ones are promising new drug targets for designing new drugs. So I talked about how we can integrate proteomics and metabolomics data and create these context-specific models, how we can interpret these high-throughput data using these. The next finally I want to talk about how we can use transcriptomics data to create context-specific models. So using transcriptomics is probably the most hardest to do because when you overlay transcriptomics data with the metabolic model, you're assuming that the transcript levels correlate with protein levels then reflect enzyme activity, which is usually not the case. And so a lot of people have tried using transcriptomics data and combine that with metabolic modeling. Usually it doesn't work that well. But transcriptomics data is the most abundant and easiest to generate. And so it's always like a big challenge to interpret transcriptomics data with these models. So I'm going to talk about a mechanistic approach that we developed to interpret transcriptomics data. The way we do it is by identifying the molecular factors that are involved in regulation, and then we try to use that to mechanistically model transcriptional regulation of metabolism. So, most of you would have learned in basic molecular biology about the lack of PROM. So, the lack of PROM basically senses the presence of lactose in the environment, and it then turns on genes required for either lactose metabolism and transport. So, even in a bacteria like E. coli, not all the thousands of reactions that are present encoded in the genome are active at any given point of time. So, most of them are turned on or off depending on the environmental conditions by transcription factors and signaling molecules. So, we want to model this mechanistic regulation of metabolic pathways. And one of the first methods that can combine transcriptional regulation with metabolism was called RFBA, or regulatory flux balance analysis. And the way RFBA works is that you manually write rules that connect the activity of transcription factors to metabolic genes. So, for example, in the lack of PROM case, we might write a Boolean rule that says if lactose is present, transcription factor X is on, and if transcription factor X is on, then it will turn on genes required for lactose metabolism. So, once you write these detailed Boolean rules, you can then use that to simulate the metabolic network. For example, let's say you want to predict what happens when you knock off this transcription factor, then you would go back and evaluate the Boolean rules. That would say that, for example, if this transcription factor is not active, then these two genes are not active, and maybe this gene 3 is active. And then you would use the model and overlay this information onto the metabolic network, and then you would turn off these reactions. And then you will run the metabolic modeling again and figure out what happens when you turn this gene on or off. So there are two main issues with this. One is that gene regulation is generally not binary. So it's very rare that you would find cases where when you knock off a transcription factor or turn it on, a gene becomes either completely active or completely inactive. It's usually more nuanced. Another issue is that you need to write these rules manually. It's fine if you're playing with a toy model, but for systems like even E. coli or yeast, where there are thousands of regulatory interactions, it's really hard to write these Boolean rules manually. So we developed a new method called PROM, which stands for probabilistic regulation of metabolism, that addresses both these issues. In PROM, we represent these transcription factor gene interactions as probabilities. So instead of a gene being completely on or off, we can have more nuanced regulation. A gene can be on at 30% of its activity or 50% of its activity. And the way we quantify these probabilities is by using gene expression data. So PROM, in an automated fashion, looks at hundreds of microarrays and then figures out what happens when a transcription factor is off, and then figures out what the connection is between the activity of a transcription factor and the activity of its target gene. For example, in this case, PROM might say when this transcription factor is off, this gene has a 26% probability of being on. So we can use these probabilities into our modeling. So rather than turning off genes completely on or off, we can have more nuanced regulation of metabolic fluxes. So this is how it pans out in the systems level. So we use PROM to build a model for E. coli, where we have more than 2,000 metabolic reactions, more than 1,700 regulatory interactions, and use data from more than 900 microarrays. So then, by combining these different kinds of data, we can ask complex questions like what happens when I knock off a transcription factor, or what happens when I change the metabolic condition, and how does that affect the growth of the cell. So we can build really large models with this. So using a manually curated approach, the RFBA, we can only have models like a few hundred interactions, whereas with PROM we can have interactions like more than 1,500 interactions. And this also improves our accuracy of the model predictions. So in this case, we tested PROM using data from more than 1,800 different growth phenotypes. So what this means is that we knocked off different transcription factors and we exposed the bacteria to over 125 different growth conditions. This may involve different carbon sources, different nitrogen sources, are a combination of gene knockouts and different carbon sources. And PROM was able to predict the outcomes with 85% accuracy in over 1800 different conditions. We can also make predictions quantitatively. So we can figure out what happens to the cell when you change, for example, the levels of oxygen, or you can knock off different genes. For example, in this case, RK is an important regulator of hypoxic response in E. coli. So we knocked off multiple transcription factors and changed the metabolic conditions. And asked the question, can PROM accurately predict what happens to the cell when you do these genetic perturbations? And we found a very good correlation between the actual experimentally measured growth rate and the ones predicted by PROM. So PROM can be a great tool for engineering the cell. For example, if you're a metabolic engineer and you want to overproduce ethanol, or you want to produce a biomolecule of interest, you can now use PROM to identify a set of gene deletions or transcription factor deletions to engineer the cell. And you can also use it for drug discovery. For example, in this case, using PROM, we built the first model that combines transcriptional regulation metabolism for tuberculosis. So this is the pathogen that causes TB, and we built a PROM model for this, which involved more than 1,000 metabolic reactions, more than 200 regulatory interactions. So it's not as big as the E. coli model because we didn't have that much data on this. But despite the fact that we had very little data, we were able to predict with around 95% accuracy the outcome of transcription factor deletion. So we can predict what happens when you delete important transcription regulators and how that impacts the growth of the bacterium. PROM only made one error, that was for this gene KDPE. It's involved in ion metabolism, and the reason we were able to incorrectly predict this outcome was because ion metabolism was not part of our network, and so we made this incorrect prediction due to lack of annotation in the metabolic model. So this was published in 2010, and a lot of other groups have now taken this PROM model and expanded it. So they have built larger models for tuberculosis. They also use this to understand hypoxic response for MTB. So adaptation to hypoxia is important in this context because bacteria needs to survive in intracellular conditions where there is lack of oxygen. And so other groups have also identified drug targets using PROM. So far I've talked about how we can take different kinds of data, overlay into this model, and find something really cool with it. But normally when you do this for your own project, you might face a lot of challenges. For example, you may not have data for building these models. Another common but really challenging issue is that sometimes your transcriptomics data might say, might predict X, whereas it might conflict with what your metabolic data is saying. So there may be conflict between different kinds of omics data. And so I want to talk about two main challenges that we face when we build these models and how we are trying to address them. So for example, let's take PROM again. To build PROM model, you need metabolic networks, obviously. You also need gene expression data to quantify the relationship between transcription factors and target genes. And you also need the regulatory network. You need to know what transcription factors control which genes. It turns out that there's a lot of data on metabolic network topology for most organisms. There's also a lot of cheap microarray data. It's easy to generate, so even if you don't have it, you can always measure gene expression. What's really hard to get is this third one, the gene regulatory network. So I'm going to talk about in cases like for non-model organisms where we don't have that much data, how can we address this issue? So we developed a new method called ASTRIX that can build regulatory networks on the fly using gene expression data. So this is fairly abundant, so we can use this data to sort of get like a guess regulatory network that you can then use in your subsequent modeling. So the way ASTRIX works is that it takes in transcriptomics data, and then tries to come up with a mathematical model that connects the expression of a target gene of interest to its corresponding transcription factors. The way it does it by using two different statistical techniques. The first is mutual information. So what mutual information does is it measures the correlation between two genes' expression levels. So let's say these two genes have very similar expression pattern across multiple conditions, then ASTRIX would say that there is an interaction between a transcription factor and a target gene. So once it identifies these interactions, it then quantifies it by using an algorithm called least angle regression. So using least angle regression, you can write a mathematical equation that connects the expression level of different transcription factors to the expression level of the target gene. So then using this approach, you can identify what transcription factors are regulating a target gene, whether they're going to activate it or repress it, depending on the sign. And you can also identify which ones are more important, depending on the value. Like the magnitude of these coefficients can tell you that transcription factor 1 is more important than transcription factor 2. So using this approach, you can get a quantitative model for the regulatory network, which you can then use it for subsequent network level analysis. So we applied this for a lot of non-model organisms. So for E. coli or yeast, there's already a lot of binding data that can use for building these regulatory networks. But for other less studied systems, it's really challenging. So we have applied it to a lot of different organisms, starting from honeybees to fish to even mouse. For example, so one important thing about ASTRIX is that it creates what's called context-specific regulatory networks. What this means is that it does not give you a model for mouse. It gives you a model for mouse, in this case, or mouse responding to an intruder threat. So, for example, when you expose mouse to a foreign intruder, then there's change in brain gene activity. So we were able to use ASTRIX to identify the set of transcription factors that were involved in regulating mouse response to this intruder threat. We also build models for honeybees. For example, honeybees are showing aggressive behavior or foraging for new resources. In all these cases, we can identify transcription factors that are active in specific contexts. So that's one example of when you have lack network data, we can infer just using microarray a regulatory network, which you can then use that for subsequent modeling or just by itself. So I want to talk about the next example, next challenge rather, that is what happens when you have conflicting data and how can we resolve this. So we're going to talk about a new approach called GEMINI that we developed that can help resolve inconsistencies between different data types. So let's go back to the example of PROM again. So let's say you did a ChIP-seq experiment and you found that the transcription factor 1 is predicted to turn off a specific gene. So if that was the case, then you can ask using PROM what happens when you knock off this transcription factor. Let's say your data would predict that if we turn off this transcription factor, then this enzyme will also be turned off. And what the algorithm predicted is that in that case, the cell would not survive in a certain condition because it needs this reaction to be active to metabolize an important metabolite. And then you do this experiment and you find that the experiment does not agree with your model prediction. So what do you do now? So GEMINI helps to resolve this inconsistency. It tries to identify the data type that is most likely to give this error. So for example, in this case, the error might be due to 3 different reasons. One is that you did your experiments wrong, which is highly unlikely because growing a cell and measuring viability is something very easy to do. So you can't mess that experiment. So then you can ask, it's either going to be the transcriptional regulatory interaction or the metabolic network. So if this falls into a central metabolism, which has been studied for hundreds of years, then it's most likely that the error is due to the transcriptional regulations and metabolism. So then GEMINI can then figure out among the thousands of regulatory interactions, which ones are most likely to cause this error. You can do this iteratively, and then, for example, let's say you resolve one error, and then you do a new experiment and you find a new error. Then GEMINI can go back and find the source of this inconsistency in the data. So we applied GEMINI to yeast, where we had more than 20,000 regulatory interactions, and most of this data was pretty noisy. This was from different sources like ChIP-seq data, RNAi, gene knockout, motif search algorithms. And so what we found was that when we did use PROM to predict phenotypes, we found a lot of errors that are what we call as inconsistencies between model predictions and experimental observations. But using GEMINI, we were able to identify where the source of error is. We were able to find that these 3 transcription factors and their network were responsible for thousands of errors that we observed during our modeling. So then we can focus on our experimental curation on these 3 regulators alone, and we can resolve thousands of inconsistencies that we observed during our modeling. So another interesting thing is that because these different kinds of data were from different sources, and what we found was that data from ChIP-seq and gene expression were sort of high confidence, and data from motif search algorithms were the ones that are most likely to show these errors. And most of this data were from using data from motif-seq algorithms. And so probably the advice is that when you're building these models, try to use either ChIP-seq data or data from RNAi knockoff experiments to build these regulatory networks. So Gemini sort of represents like a new way of modeling. So throughout this talk, I talked about how we can overlay different kinds of data onto these network models. So these are models built from prior knowledge. These are mechanistic models, and you overlay new data onto it and try to infer what the metabolic activity is. Whereas ASTRIX is a data-driven algorithm. It just uses statistical correlations between two different genes' activity to figure out the network architecture. And Gemini is sort of it's a bridge between these two different kinds of models. So it takes in statistical data and then overlays that onto mechanistic networks and tries to identify inconsistencies between different data types and tries to resolve these. So it tries to build more larger hybrid models by combining different kinds of modeling frameworks. So to summarize what I've said so far, I talked about how we can use these genome scale models to interpret different kinds of data and how we can create context specific models by using different kinds of omics data and overlaying that to the model. Also talked about some challenges that we face. And for the next 2 minutes or so, I'm going to talk about some interesting directions where we are taking this in the future. So one power of these models is that we can do all sorts of perturbations to the system. So I talked about how we can use PROM to predict growth phenotypes in over 1800 different conditions. We can do gene knockouts, change environmental conditions. So using these algorithms, you can sort of figure out the entire combination space of genetic perturbations that you can do in a system. And so in our lab, we are now trying to use these algorithms to design combination therapies for infectious diseases. And so there's a huge problem right now in that for many of infections like tuberculosis or staph aureus, we do not have new drugs to treat them. And so one common way to target drug-resistant pathogens is by using a combination of existing drugs. So by using combinations, you can fight resistance, and you can also enhance the potency, and you can also increase the spectrum, that is the kinds of pathogens that are being targeted with the same set of combinations. But there is a challenge in this, in that, for example, even though many of the antibiotics target distinct processes, for example, some drugs target cell wall synthesis, some drugs target DNA replication, some target translation, but they're all sort of part of the same cellular network. And so adding one drug, for example, a cell wall inhibitor with a DNA synthesis inhibitor, might actually interfere with each other's action. And so we can use these models to figure out what two drugs to give together, and that might work very well with each other, which is called synergy. But you can also identify drugs that interfere with each other's action or they are antagonistic. So using these models to build and identify, search through like thousands of combinations to identify which ones are most effective when added together. So we are working with the David Sherman lab in Seattle, where we're trying to test new combinations that we are predicting for tuberculosis. And the way we do it is by we are taking different kinds of data and then combining them to build a model. So in this case, we are using tuberculosis data exposed to different drugs, and then we're also using data where we know the effect of adding combination of drugs to the pathogen. We're also using data about the cellular network, that is which transcription factors regulate which genes, and we know what genes respond to which drugs. We also know how drugs and drugs interact. So putting all these three data together, we can now predict for new drugs what happens when you combine them either in combination of 2s, 3s, or 4, and then you can also identify which transcription factors mediate these interactions. We can identify rational ways to enhance synergy between drugs or reduce antagonism between different classes of antibiotics. Another interesting direction which we are taking this by building more comprehensive metabolic models. So I talked about how we can model transcriptional regulation of metabolism. So moving forward, we're also trying to identify, develop new methods for identifying regulation by small molecules like metabolites, and also how metabolism impacts the epigenetic state of cells. For example, I talked about in stem cells how metabolic enzymes affected histone methylation. Similarly, we can also figure out how histone acylation is affected by the metabolic state of the cell and how that impacts the expression level of enzymes. We can understand metabolism more comprehensively. With that, I'd like to conclude and thank various people I've collaborated with over the years. So all the stem cell work was done in collaboration with the George Daly lab, and Jin Zhang led the study. And then the T-cell work was done in collaboration with Judy Lieberman's lab, and Farooq Dodiwala led the experimental effort on this. And then the TV stuff, which I briefly mentioned, was done in collaboration with David Sherman's lab in Seattle, and Sriram led the study on that. I also want to thank various people that I've worked with over the years. I did my PhD at the University of Illinois, and then the Institute of Systems Biology with Nathan Price. I also collaborated with Gene Robinson's lab for the neurogenomics work, and then I moved to Harvard and I collaborated with Jim Collins on the stem cells and antibiotics work. I also want to thank all the funding sources, and thank you all for your attention.
That was great. We have time for a couple of questions.
I have two questions. The first, in the network reconstruction by artist, the initial correlation is established by mutual information, which does not decouple direct from indirect correlations. But in your pipeline, you explicitly say that direct correlation, which I don't understand how we can establish. The second, in the link 28 knockout and naive versus prime pluri-cell, why does the most significantly change metabolite are GTP and ATP, which are very fundamental to the cell?
Yeah. So for the first one, yeah, I did explicitly mention direct versus indirect because actually in our algorithm we have a way to differentiate from the two. So we use what's called use the triangular approach. Where, for example, let's say gene A has a very high image information gene B and gene C, and we know the correlation with B and C. We use the triangular approach to prove out the weakest edge between them. So using this approach, we can identify the direct regulation from the indirect ones. So it's called the data processing inequality approach. I don't know if you're. Yeah, exactly. Yeah, exactly. Yeah. So if they're identical, we can't. But if there's one that's lower than the other two, then we assume the other stronger ones are the direct ones. And so it's been shown for building a B cell regulatory network that it's able to differentiate direct from indirect. I mean, it's not perfect, but that's the statistical noise that you get using that data. And for the second question, we do find that ATP, GTP are purines, and we find that pure metabolism is impacted when you perturb folate metabolism. And so that's why we see ATP and GTP, but not UTP and TTP that are changing. So only purines are changing, and that's what the model predicted too, and we were able to validate that using both flux tracing and metabolomics.
Hey, that was really great. A lot of really nice tools. So I was wondering if you could comment on the relative utility or accuracy of using different types of experimental data, like time course metabolomics versus transcriptomics versus proteomics.
So for the gene knockout experiment with the NCI60 cancer cell lines, metabolomics gave the highest accuracy. Proteomics was statistically significant. Transcriptomics was not helpful at all. In fact, when we try to combine it with others, sort of reduce the accuracy. So we don't know if it's only for the cancer cell lines or even for the stem cell case, using transcriptomics did not provide a good accuracy in prediction. So I would recommend using metabolomics or proteomics.
Time course metabolomic data would all have been targeted metabolomics.
Yeah.
So can you predict like how it might behave when if you're using like an untargeted data set, or can you not?
I mean, as long as it's time course and you're measuring the same sort of metabolites over time, that's fine. But I'm assuming in that case, you'll have a lot of missing data. I mean, in our case, what we found was that we randomly simulate that. So we had around 100 metabolites that we can measure consistently. We just had 50, then our accuracy dropped significantly. So I guess there are some few key important ones that the algorithm needs, and others are probably in peripheral part of the network, and so they don't give the same amount of information as like glucose or other central metabolic metabolites.
Okay, that's great. Let's thank Sriram again.